Hi, everyone. Thank you very much for joining. My name is Alexandre Bodo. I'm a pediatric rheumatologist in Lyon, France, and I'm very happy to co-chair the session with Jad Cognard, a junior pediatric rheumatologist in my team also. We may wait 1 or 2 minutes before you can join the, the webinar. So we're lucky enough to have a great speaker today, and we'll discuss about NFkB-opathies. New field of disease, let's say, and I think it crossed the interest of all the stream of the RITA community. So welcome everyone. I think, yes, we can wait for 1 or 2 minutes. Normally, there is more than 100 people who are supposed to join the meeting. It's typically the ideal time, so you can take a sandwich or coffee and learn more about immunology and translational immunology. So all these sessions are recorded, so you can also see it again after the talk. When you go for work, when you're cycling, or in the metro, you can still listen about this Learn with Rita webinar. We are already 60 connected. Welcome, everyone. Well, it's 12, so we have just a short hour. Maybe we could start. So welcome everyone again. Thank you for joining us for this Tuesday Lunch with Rita webinar on NFkB-opathy. We want this to be as interactive as possible. So you can use the Q&A and we will cover the question at the end of the talk. So use the Q&A please and don't hesitate during the different presentation. And we're going to start with Professor Bodo Grimbacher. So welcome, Bodo. So Bodo is the scientific director of the Center for Chronic Immunodeficiency in Freiburg, vice director of the Institute of Immunodeficiency, and he's really famous. No need to introduce him. So trained in rheumatology, clinical immunology. He's an expert in the translational immunology field, internationally recognized for his contribution and outstanding work, especially on NFkB1 haploinsufficiency, as well as STAT3 haploinsufficiency, as you know. So we are very, very proud to welcome you, Bodo. Thank you for joining. And the screen is yours.
Thank you very much, Alexandra. Thank you for the kind introduction, also for inviting me to speak. Can you see my slides moving? No, you can't.
We can see the first one and the second one. Yes,
Okay,
perfect.
cool. So I would like to start with my disclosure slide, and this is just to show that not only by public funding, but also by pharma funding, we are trying to develop novel treatment management procedures in the field of inborn errors of immunity. I work in the CCI, the Center for Chronic Immunodeficiencies in Freiburg, Germany. And the mission of this institute is not only to recognize patients and diagnose them, but also to understand their pathophysiology, their underlying pathophysiology, and then to develop novel treatment strategies. And today's topic, NFkB1 mediated diseases, are at the center within ERN-RITA. And that is because ERN-RITA not only encompasses the primary immunodeficiencies, but also the rheumatological diseases, the pediatric and the adult ones, and the autoinflammatory diseases. That is, although I work in primary immunodeficiencies, and there where most of the patients come to Freiburg with a suspicion of an immunodeficiency, many of these patients also, in addition to the defective immune system, have autoimmunity, have autoinflammatory diseases, as I will show later. However, the patients coming to Freiburg are mostly immunodeficient patients. And in this chart, you can see that half of those patients showing up with immunodeficiencies actually have an antibody deficiency. So they suffer from hypogammaglobulinemia, and their antibody response is not perfect. And because of this, they suffer from recurrent infections. When you look at those patients with antibody deficiencies, primary antibody deficiency, PAD, then we can these days diagnose about 25 to 30% with a monogenic molecular cause. So you can see here how these monogenic patients split up in different monogenic traits. In Freiburg, the most frequent one is CTLA-4 haploinsufficiency, but you can see also on the bottom of this chart that NFkB1 and NFkB2 monogenic causes are another big chunk of these patients leading to antibody deficiency. We have first published mutations in NFkB1, one factor of the NFkB signaling pathway, back in 2014. And the phenotype of the first family described with a genetic mutation in this gene had CVID, common variable immunodeficiency, which is the most frequent antibody deficiency in humans. However, it didn't take long. 1 year later, patients with a combined immunodeficiency, so B and T cell defect with NFkB1 haploinsufficiency were described. And in the same year, patients with autoinflammatory disease phenotypes and mutations, heterozygous mutations in NFkB1 have been published. So it became clear from those first years that NFkB1 mutations can cause a very heterogeneous clinical and immunological phenotype, encompassing autoimmunity, autoinflammation, Behçet's-like vasculitis, familial necrotizing cellulitis, EBV-associated lymphoproliferation, chronic inflammatory bowel disease, and the susceptibility not only to bacterial, but also to viral and even opportunistic infections. So I'm going to show you a couple of pictures, and this is to show you how NFkB1 mutations may manifest. They may manifest with skin disease. Rosacea is a very characteristic feature. They may manifest with alopecia, and alopecia may be areata or totalis or even universalis. It may manifest with interstitial lung disease. It may lead to clubbing. It may lead to lymphoproliferation. You can see here this huge enlarged spleen, even bigger than the liver. But also the liver can be increased. It leads to fungal infections, especially by Candida. And mostly feared, it leads to pyoderma gangrenosum. So picking all these or putting all these phenotypes together, back in 2020, Tiziana Lorenzini, who now works in Zurich, put together a compendium of what the phenotype looks like. So if you are interested in how the phenotype may manifest, I advise you to read this Journal of Allergy and Clinical Immunology paper. But virtually many organs, almost all of the organs may be affected. The disease may also pass the blood-brain barrier and cause neurological complications. So what is the pathway we're talking about? We're talking about NFkB signaling, and NFkB signaling, honestly, is not only a pathway, it's a network. And we have to differentiate between the canonical NFkB signaling and the non-canonical. Tom later will talk about the non-canonical pathway because non-canonical pathway is mediated by NFkB2. NFkB1 is the canonical signaling pathway, and it's important to teach you that the gene produces a protein called P105. So the P105, the long protein, is a precursor protein, which is proteasomally processed to P50. P50 then complexes in the cytoplasm with its partner in crime, the REL-A, which is also called P65, and an inhibitory protein called IKB-alpha. This trimer, P50, P65, and IKB-alpha is retained in the cytoplasm as long as IKB-alpha is there. If IKB-alpha is degraded by phosphorylation and ubiquitination, then the active transcriptional dimer, P50, P65, can locate with a membrane localization site into the nucleus and initiate gene transcription. So the P105 is the full-length protein encoded by NFkB1, but it needs to be proteasomally cleaved. As you can see here, the C-terminal part, the C-terminal half of the protein is clipped off to release the P50, which is the DNA binding transcription factor. This is important to know because all the C-terminal mutations in NFkB1 obviously may exert a different effect than mutations affecting the P50 itself. So what is the phenotype we would expect if you have an NFkB1 haploinsufficient individual? So that means haploinsufficient means one allele is not working or does not produce a protein which is detectable or biologically active. Because NFkB1 signaling, or the canonical signaling pathway, is known to be involved in inflammation, survival, and proliferation, haploinsufficiency would suggest that a patient with mutations in NFkB1 leading to insufficiency may have reduced inflammation, reduced survival, and reduced cell proliferation. However, with the pictures I just showed you, I hope it became clear that the phenotype we observe in our patients is hyperinflammatory. For instance, best example is the pyoderma gangrenosum, and lymphoproliferation. You remember the big spleen and the big liver I've showed you. They also have big lymph nodes. So we have an haploinsufficient NFkB mutation, which leads to hyperinflammation and hyperproliferation, at least of lymphocytes. So this may be explained by the fact that, as I said, NFkB is not a signaling pathway. It's not from the receptor just down to the nucleus and that's it. NFkB signaling is a signaling web, a signaling network where many feedback mechanisms are implemented. And the mutation, obviously, the haploinsufficiency mutation, obviously leads to a greater effect on the negative regulator of NFkB signaling than it does to the positive feedback mechanisms. So we have shown that in unstimulated B cells, in naive and switch memory B cells, but also in stimulated B cells in all the patients and also in the affected mutation carriers, that the negative regulators in these cells are less expressed, and therefore we expect probably a pro-inflammatory loop in this signaling pathway in patients with haploinsufficiency mutations. However, as I said, there are also some mutations which are on the C-terminal side of the protein, so affecting P105 but not P50. So for instance, this is a stop codon after the cleavage site, and this stop codon produces a protein which is P50-like. So one of the two alleles of these patients produces the wild type, and it needs to be clipped off the C-terminal half, but the mutation produces an allele which produces a P50-like protein. It doesn't need to be processed, so it's immediately P50. So this one allele which produces a P50-like protein should lead to an increase of P50 molecules in the cytoplasm of patients. How can I then explain that precursor skipping mutations, this is how we call those mutations, shouldn't they produce a different phenotype than the haploinsufficiency mutations, where I just said that there is less P50? Well, explanations may be that P50 not only dimerizes with P65 and other REL proteins, but it also homodimerizes. And as a homodimer, P50 homodimer also bind to the P50 DNA consensus sequence, the NFkB1 consensus sequence on the DNA, but functions as a transcriptional repressor. Because P65 or the REL proteins are needed because they confer the transcriptional activation signal. So if you have no P65, no REL-A, no C-REL, no B-REL, there's no transcriptional activation, and therefore P50 homodimers or P50, P52 heterodimers will lead to a transcriptional repression. So on the left side, it's a wild-type situation. On the right side, it's a wild-type mutated situation with a P50-like mutation. And they have, of course, an increase in P50 in the transcriptional activating, but they also have an increase in the homodimers without the transcriptional activation side, leading to a repression of transcription. So in the end, we end up, depending on where the mutation is, with 8 different types of NFkB1 variants. And this is very important to note, that depending on where the mutation and how the mutation looks like, it may lead to a different biological effect. Interesting to us, however, and I just had a medical student finishing her thesis, the phenotype of all those 8 different variant classes are very, very similar, and statistically, at least with the numbers we have collated, cannot be differentiated. So it does not matter where the mutation is, but it does matter that the mutation has a biological effect. So here again, a schematic scheme of P105, and the proteomal processes leads to a clipping off of the C-terminal part, half of the protein, leaving intact the P50, and the P50 then translocates it together with REL-A into the nucleus. Now, that clipping off defines that all the mutations which are behind or after this clipping site cannot be functionally tested if you test P50 function, because these mutations will be clipped off. And this can be seen by our publication in the Journal of Experimental Medicine, that all the mutations following position 433 returned in the functional test, where we test the P50 part of the protein, were tested functionally normal. You can see here in the front 1.5 rows of the results that there are some mutations which impair the biology of P50 signaling, but if you see a full bar up to the number 10, then the mutation does not impair the functional, and has no functional impact. And you see that after row 1.5, all the functional effects are normal. That doesn't mean that that does not mean that the mutation doesn't have a functional effect. It just means that the readout we used for that assay does not demonstrate the functional impairment of these mutations. Therefore, together with Manfred Fliegauf in the lab, we have developed 4 different readouts for testing the integrity of NFKB1 signaling. One is obviously we can measure how effectively P105 is translated into P50 by protein processing, and we do that by Western blotting. Second is we can measure how efficiently P50 is then translocated into the nucleus for gene transcription. We can then also measure by EMSA the effectiveness of the mutated P50 to bind to its DNA recognition sequence. And last but not least, we can measure target gene expression by reporter assays. So these are examples. Here, the version number 2 shows how effectively these mutated proteins can translocate into the nucleus. And you can see that there are some mutations, for instance, the Y3500C mutation, where the protein expression itself is prohibited, despite that is a point mutation. But we can also see that the exon 8 deletion also leads to a reduced expression of the protein. It can be expressed in the cytoplasm, but when it then translocates into the nucleus, it leaves those funny clumps. We can measure the by Western blot, and you can see here that the double mutant, as shown here in blue, has a reduced Western blot signal when compared to the single mutant or the wild type. We can look at DNA binding with the EMSA shown in the middle panel, and there you can see here that the double mutant in this patient has a reduced DNA binding. And we also can see that the reporter assay of this double mutant is reduced when you look on the right-hand side of my slide. In the second half of the talk, I will actually talk more about the clinical findings in these patients. And I take advantage of a great data collections of colleagues of mine listed here into a database called GENIA. GENIA is a genetic immunology advisor database, which is basically database collection like OMIM, but focusing on genes causing primary immunodeficiencies or inborn errors of immunity. So this project has 3 steps with data acquisition, GENIA documentation, and data analysis. And out of this, I need to close the window. Out of this, following information became clear. So first of all, the paper was first published in 2014, the gene description. So you can see that following this, many more publications on NFKB1 were shown. And you may wonder why there was a publication on NFKB1 in 2006 and 2000. It's because of the initial family, which was published in 2014. The linkage analysis was published in 2005, and the first clinical description of the family was in 2001. So basically, it took us 5 years at the time to perform the linkage analysis and know the stretch where the mutation must lie. And then it took another 10 years, unfortunately, to identify the mutation in this family, which was then showed to be haploinsufficiency of NFKB1. In these published papers, there are almost 300 confirmed NFKB1 patient carriers, and there are additional 440, roughly, healthy family members. This slide needs to make us aware that in these publications, some of the patients have been actually published several times. So if you just count the numbers of published cases, you will be misled, because there are patients which are actually published, the very same patients, up to 7 times. And this made the GENIA research clear that many of the patients have been published twice, 3 times, or even 4 or up to 7 times, the very same individual. Second of all, these publications made clear that NFKB1 has a reduced penetrance. So only 74.2% of the mutation carriers are actually affected. There are some mildly affected, but also very importantly, 22% of mutation carriers are unaffected. So we're talking here about a disease with clearly reduced penetrance. The other thing which made this collection of data made clear that the age of onset is very, very variable. So we have many age of onset in childhood. We have a lot of age of onset during adolescence and adulthood, but there's also age of onset beyond age 60. So we have no clear idea why that is in the field of NFKB1 research. However, it is clear that once you manifest it with the disease, life expectancy is clearly reduced. I would like to ask you to focus on the right-hand side of the slide, where you can see that not significantly, but possibly males are more severely affected and die earlier than females. They may die in childhood. So there are 3 patients we lost already in childhood. But many patients are deceased around the ages of 40, and very few, however, also make it until the age of 80. And those are probably the ones who have not manifested with the disease and die of a natural cause, because the penetrance was, there was no non-penetrance. Now, the question was, is the disease penetrance a factor of age? You may argue that the older you get, the more likely it is that you manifest with the disease. But as you can see in this analysis, that is not the case, because even in the 75 to 85-year-old patient mutation carriers, 25% were healthy, so non-penetrant. So it seems to be that there are other factors keeping patients healthy despite having a pathogenic NFKB1 mutation. So the big question is now how to determine NFKB1 disease activity. We do need that activity measurements to come up with drugs to be proposed for the management of these patients. I can only say that a drug worked if I have pre and post disease activity measurements. And because it's a multi-system disease, we need a composite score to measure disease activity in this disease. So Katharina Thoma from my group collected 8 publications with 230 NFKB patients and drafted a score of 41 score parameters depending on the phenotype described in those 8 publications. And then she gave a weight of how important that phenotype was in these patients. And she did a leave one out analysis and came up with 21 out of those 41 parameters, which are significantly reliably telling us about the disease activity. So we now have, and this is currently a paper we have submitted, there is a 21 parameter disease activity score, which measures as a composite score all different or many different organ involvements we see in NFKB1. And many of them I've shown you clinical pictures. I'd like to acknowledge our work in NFkB1. Of course, because these patients are rare, we rely on the collaboration from all over the world. Currently, my colleague Cleona Murray is collecting additional clinical data to use that disease activity score to measure which drug has possibly helped these patients and which one has not. We already know that in some patients, anti-TNF works. We have knowledge about anti-IL-1 working. These two make sense because they feed into the NFkB canonical signaling pathway. But also we need to know whether, for instance, JAK inhibition is superior to these other possible treatment module. I'd like to thank you, my group, shown here, all the people who helped putting together working on it. I'd like to highlight Manfred Fliegauf, who does the most of the molecular work in our group, and the medical students who helped putting together all the clinical data. Thank you very much.
Thank you. Thank you very much for your fascinating presentation and for sharing your great expertise with us. So please don't hesitate to ask if you have questions in the chat, and we will answer them at the end of the webinar. And so now we will have the second talk of the day by Tom Le Voyer, who is a clinical immunologist, and he conducted his PhD and is now a physician scientist at the Laboratory of Human Genetics of Infectious Disease in Imaging Institute in Paris. And his research focuses on the genetic and immunological determinants of severe infectious disease through the study of inborn errors of immunity and their autoimmune phenocopies, particularly inherited defects of the alternative, so non-canonical NFkB pathway. So we are very pleased, Tom, and the screen is yours.
Thank you very much, Jad, for this kind introduction. I will share my screen. Can you see it full screen?
Yes.
Yes.
Okay, so the second part of this session will focus on inborn of NFkB2 and a bit more generally on the alternative NFkB pathway. So this NFkB pathway is formed by NFkB2 transcription factors, which are essential regulator to control the transcription of several genes, which are involved in survival, proliferation, and immune functions. They are involved in both adaptive and innate immunity, which can be mediated by both hematopoietic or stromal cells. So these NFkB transcription factors can be delineated in two different pathways or network. The canonical NFkB pathway, which is mostly involved in the inflammatory responses with early onset, and the alternative NFkB pathway. These two signaling pathway differs from their signaling components and their biological functions. The alternative NFkB pathway is activated by the activation of upstream TNF receptors, and then this will lead to the activation of the upstream kinase, which is called NIC, and NIC will activate EKK alpha, and then will lead to the activation of NFkB2 through its precursor P100, who will lead to the degradation of the C-terminal part, similarly as the NFkB1 activation described by Bodo. And this C-terminal part will be degraded into the proteasome to give P52, and P52 is the transcriptionally active form of NFkB2. P52 will then heterodimerize with its main partner, RelB, and this heterodimer, P52-RelB, will translocate into the nucleus and act as a transcriptional factor to regulate transcription of targeted genes. And the function of this alternative NFkB pathway have mostly been described or initially in mice, and has been described to be involved in lymphoid organ development, development of thymic medullary epithelial cell within the thymus, and the development of B cells and the survival of B cells. So the study of inborn of immunity have led us to understand what are the essential role of these different components of the canonical and alternative NF-κB pathway. And regarding the alternative NF-κB pathway, there are several inborn of immunity caused by monogenic defects involving each of or several of these components have been described. 3 of them cause a combined immunodeficiency caused by homozygous deleterious mutations in NIC, REL-B, or EKK alpha, and other have been described to cause predominantly antibody deficiency in patients with heterozygous variant in NFkB2 or TRAF3. And we will focus a bit more on NFkB2 during this talk. So to understand what and the consequence and the mechanism underlying this inborn of the alternative NF-κB pathway, we need to understand how NF-κB2 works. Actually, NF-κB2 is the gatekeeper of the activation of the alternative NF-κB pathway. And this alternative NF-κB pathway is strikingly different from the canonical NF-κB pathway by some specificities. First, the alternative NF-κB pathway is an inducible, have an inducible activation. It's not activated at basal level, but it needs to be activated by stimuli. The second characteristic is that this pathway has a slow and sustained kinetics of activation. It's activated after 24 to 72 hours of activation in vitro. And there is a high cell type specificity to activate this pathway, and it's mostly in B cells and in stromal cells, as we described before. And NF-κB2 is present as a precursor, which is a protein that is shown on the top left here. And this precursor is when the alternative NF-κB pathway is activated, will lead to production of P52, and P52 is the transcriptionally active subunit. P52 will heterodimerize with REL-B and translocate into the nucleus. That's the positive and transcriptional activity of NF-κB2. But the transcriptional, the precursor P100 acts the negative regulator, and this precursor is present in the cytoplasm when the pathway is at the basal level before the activation, and it inhibits the pathway to prevent its spontaneous activity. And P100 binds REL-B and prevents its activation at the basal level. So it's a negative and inhibitory activity, which is called EKB delta for inhibitor of κB delta. And so far, 3 different forms of autosomal dominant NF-κB2 defects have been described, and they correlate with the different functional consequence of these alleles reported in patients by in vitro functional assay. The first group is caused by variants which are cause loss of function of the P52 transcriptional activity and the loss of function of the EKB delta regulatory activity. So these patients are heterozygous for a complete loss of function variant, which cause NF-κB2 haploinsufficiency. The second group is caused by variants which truncated this P100 precursor and lead to its spontaneous translocation into the nucleus, at least in vitro. And these patients have P52 gain of function. And the third group is caused by heterozygous variants located in the C-terminal part of NF-κB2 in regions which are critical for the processing of P100 to P52 and its phosphorylation by NIC. And these patients have a combined defect in terms of NF-κB2 activation. First, because this mutation caused a mutant P100, which is unable to process into P52, so there is less P52 produced, and it maintained this mutant P100 in the cytoplasm with an increased EKB delta activity, so they also prevent RELB and P52 to be functional. And it's what we can observe also in heterozygous cells of the patient. When looking at the primary fibroblast, we can observe that healthy control fibroblast stimulated with TRIP, which activate the alternative NFkB pathway, we can clearly see nuclear dots here in green representing RELB. And similarly observed in patients with NF-κB2 haploinsufficiency in orange, but in red here, mutant, there is a stronger inter-RELB activation. So these patients with C-terminal NF-κB2 variants causing EKB delta gain of function cause the more severe form of activation of this alternative NF-κB pathway. So what's the clinical consequence of these different variants? And when we recruit a worldwide cohort of patients with different form of NF-κB2 defects, actually the three different groups of NF-κB2 defects all lead to hypogammaglobulinemia with susceptibility to recurrent bacterial respiratory tract infections and autoimmunity in about 30% of them regarding NF-κB2 haploinsufficiency or P52 GOF, and relatively at a higher proportion for patients with EKB delta gain of function variants. But patients with autosomal dominant EKB delta gain of function variant caused by these C-terminal truncated or missense variants cause specific clinical phenotypes or manifestation such as alopecia, nail dysplasia, and endocrine defect caused by pituitary defects. They are also susceptible to severe viral infections. And this is what is shown here on the right. About a third of these patients with EKB delta gain of function variant have anterior pituitary defect and or ectodermal dysplasia, and about more than half of them suffer from severe or recurrent viral disease. And these manifestations were not observed in patients with NF-κB2 haploinsufficiency or P52 gain of function variant. And when we look at the viral disease, they were caused by severe form of COVID-19 pneumonia, flu, pneumonitis, varicella, herpes zoster, or recurrent HSV1 oral manifestations. And what is also strikingly different from patients with autosomal dominant NF-κB1 haploinsufficiency is that these patients with autosomal dominant EKB delta gain of function variant have an almost complete clinical penetrance during adulthood, while this clinical penetrance was much lower for patients with NF-κB2 haploinsufficiency. Regarding the immunological phenotype of these patients, so most of them, including three different forms, have hypogammaglobulinemia. They have clinical pictures of CVID patients associated with a decrease in terms of B cell count over the years. But patients with EKB delta gain of function with gain of function variants have a reduced proportion of memory B cells, Treg, and circulating TFH as compared to patients with NF-κB2 haploinsufficiency. So what could underlie this apparently severe viral susceptibility in patients with EKB delta gain of function variants? NF-κB2 is actually highly expressed in the thymus, and more specifically in medullary thymic epithelial cells, which are cells that are essential for central tolerance. And NFkB2 is predominantly expressed in mature medullary thymic epithelial cells, those that express air. And air is a central regulator of immune tolerance, which promotes the expression of peripheral tissue antigens within the thymus to educate the thymocyte and prevent their escape of autoreactive T cells into the periphery. Patients who are deficient for air function, for example, those that are that have biallelic deleterious air mutations, so patients with autosomal polyendocrine autoimmune polyendocrine syndrome 1 or IPS1, develop autoantibodies against cytokines, including against type 1 interferons in all of the cases, which predispose to their susceptibility to some viral infections. And then we test the hypothesis that these viral susceptibility in patients with incapable delta gain of function variants are caused by autoantibodies against cytokines, which neutralize the type 1 interferons activity. And we observe in these patients that the majority have indeed autoantibodies against interferon alpha-2, as shown on the left panel, similarly as patients with APS1, but it's striking contrast with the other NFkB2 defects. These autoantibodies were neutralizing in vitro and were overall identified in more than 80% in patients with incapable delta gain of function. When we look at a broader autoimmune reactivity in these patients, autoreactivity in these patients in an unbiased manner, actually at the level of this group of patients with autosomal dominant incapable delta gain of function variant, the IgG against type 1 interferons were the most enriched as compared to the healthy control in this group. And when we look at the clinical consequences of these autoantibodies regarding the clinical and immunological manifestation of these patients with incapable delta gain of function variants, all patients that had severe or recurrent viral infection actually had these autoantibodies against type 1 interferons. And among the patients with incapable delta gain of function variants, those with autoantibodies versus those without autoantibodies against type 1 interferons mostly differ from the presence of this viral susceptibility, which shows that there is a great correlation between the genotype, the clinical manifestation of viral susceptibility, and the presence of these autoantibodies against type 1 interferons. And it's illustrated here for COVID-19 pneumonia, and the patient with the incapable delta gain of function as the most severe outcome in terms of COVID-19 pneumonia, and actually with an still incomplete penetrance, as at least one patient with high titer of neutralizing autoantibodies against type 1 interferons actually did not suffer from a severe COVID-19. And this immunological pattern is also observed in other inborn of the alternative NFkB pathway, such as patients with autosomal recessive NIC deficiency and REL-B deficiency, which about 80% of those patients also neutralize type 1 interferons. And it was relatively specific for the alternative NFkB pathway because such autoantibodies were not identified in border of the canonical NFkB pathway, including patients with NFkB1 haploinsufficiency. They were also found in patients with who receive a bone marrow transplantation, and these autoantibodies actually persist over the years after the hematopoietic stem cell transplantation. We could suggest that replacement of the hematopoietic compartment does not rescue this autoimmunity. So we had access to thymus samples of these patients with complete REL-B deficiency and patients with kappa-B delta gain of function variant, those two with autoantibodies against type 1 interferons. And when we look to the thymuses, actually, there were almost absence of medulla formations and a complete absence of the green dot shown here, which represent the air expression in the thymus, strongly suggestive that NF-κB1 and REL-B are essential for the medulla formation and expression of air. And when we compare the autoantigen profile of patients with APS1 and EKB delta gain of function variant, actually, patients with EKB delta gain of function variant do not show the reactivity toward APS1 autoantigen as shown in a green dot here. They do not also have the classical peripheral endocrine autoantibodies as observed in APS1, but this autoantigen profile mostly overlap by these type 1 interferons, strongly suggesting that this alternative NF-κB pathway is essential for air-mediated central tolerance toward type 1 interferons. And more recently, other inborn of alternative NF-κB pathway have been reported and have led to additional understanding of the role of the alternative NF-κB pathway in stromal cells beyond thymus. And last year was reported patient with autosomal recessive complete LTBR defects. So it's a receptor that mostly activate the alternative NF-κB pathway. And those patients present with lymph node aplasia and functional asplenia with hypogammaglobulinemia. And these patients actually lack the formation of secondary lymphoid organs, which also suggests that this lymphotoxin beta receptor through activation of p52-REL-B is essential for the development of secondary lymph node organ. And these patients defective in lymphotoxin beta receptor have an inborn of immunity caused by a stromal defect of affecting germinal centers. And there is also one defect caused by heterozygous variant in TRAF3, which cause haploinsufficiency. And TRAF3 is a negative regulator of the alternative NF-κB pathway. So these patients which are deficient in TRAF2 have hyperactivation of the alternative NF-κB pathway and actually show a clinical and immunological phenotype, which is the opposite of what observed in inborn are affecting the activation of the alternative NF-κB pathway. These patients with TRAF3 haploinsufficiency have lymphoproliferation, organ-mediated autoimmunity, and sometimes atopy with hypergammaglobulinemia and sometimes elevated B cell number. So in conclusion, study of inborn of the alternative NF-κB pathway have led us to understand what are the essential role of this p52-REL-B heterodimer, especially in the stromal intrinsic immunity, that this heterodimer is essential for air-mediated T-cell tolerance toward type 1 interferons and LTBR-dependent development of the secondary lymphoid organs. Those patients very frequently develop autoantibodies against type 1 interferons, which underlie the susceptibility to viral disease. So these inborn of the alternative NF-κB pathway are inborn of the tolerance to self because they have autoimmunity against their antiviral immunity. With this, I would like to thank many people, first from the lab here at the Imagine Institute and our other collaborators, scientific, clinician, and the patient and their family. Thank you very much.
Thank you, Tom, Bodo. Yes, we can applaud, and that's really fantastic. And these two talks were really, really fascinating. Thank you very much. We will jump to the Q&A, so please don't hesitate to ask questions. I see that there are some already, but I should also, yes, clearly state that it's very difficult to work on transcriptional factors as you did because of this cleavage problem, because of the dimerization or homodimerization, which means it's very, very challenging to learn about this specific disease. So congratulations to both of you. So Jad, we have question, I think, in the Q&A.
Yeah, the first question is from Ben Chilito, who says, thanks, Bodo, fantastic talk. And he asks if you have any data or thought on HSCT for NF-κB1 defects.
And I think it's close to the other question from Diana Argentina, who also asked about this bone marrow transplantation, actually in both cases, and you discussed it a bit about Tom, but yeah, happy to learn more about this.
Yes, so thanks for those questions, Ben and Diana. Unlike in NFkB2, where hematopoietic stem cell transplantation will not cure the complete defect because of its role in epithelial thymocyte signaling, we do currently think that NFkB1 mutations may be cured by bone marrow transplantation, hematopoietic stem cell transplantation. This comes from the observation of currently 3 only, and I'm really surprised that it's only 3 patients whom I'm aware of who have been transplanted. Of those, 2 are children, and they have been transplanted only in the last 2 years, so the outcome is not completely clear, but it seems promising. And one 14-year-old, one currently 16-year, no, she must be older. She has been transplanted more than 5 years ago at the age of 14. And unfortunately, her disease post-transplantation was characterized by graft versus host disease, and she's currently on the similar drugs she had been for the immunosuppression prior to her transplant. So yes, she's attending school. Yes, she's doing her Abitur, and she enjoys life with her friends. But it's not like it's not that she's drug-free. So she needs to fight graft versus host disease, chronic graft versus host disease currently. Whether this is a characteristic of this disease, additional transplantation courses need to show. And again, I think the earlier to transplant, and Tom may allude to this as well, the better.
Thanks, Bodo. I don't want, Tom, if you want to comment on the specificity of NFkB2.
Yeah, actually, it's also a bit surprising, but there is even a bit more patients with NFkB2 defects who receive bone marrow transplantation, between 8 and 10, I would say. And actually, the clinical outcome is particularly severe. Most of these patients have diseased early post transplantation, and actually it may be because of this trauma intrinsic role of the alternative NFkB pathway. Regarding the other inborn of the alternative NFkB pathway, which cause combined immunodeficiency, some others have received such procedure, and actually it could lead to improvement of this combined immunodeficiency, but with the persistent, at least in some patients, of these autoantibodies against type 1 interference, we suggest that those patients still are still susceptible to develop severe viral disease. And it's 2 different aspects. Yes, sorry, Bodo.
No, may I ask a question to you, Tom? With regard to the treatment of these patients, do you think that a combined thymus and bone marrow transplant would help? And the second question is, do you actually treat your NFkB2 patients with B-cell depletion? So is rituximab useful, efficient management of these patients, or do they develop even more problems despite being on immunoglobulin replacement?
So regarding the different conditioning and of NFkB2 patient. I don't have actually the detail of this, and it's something that we trying to do and to collect patients with NFkB2 who receive bone marrow transplantation and those with NIC, RELB, and other who receive bone marrow transplantation to have a better understanding of the procedure and the clinical outcome of this patient. And we would be happy to collect more patient if there is.
But do you think if the combination of bone marrow transplant with thymus transplant, would that be a way to go?
Yes, I think that at least for a patient who receive bone marrow transplantation early life because of combined immunodeficiency, that's a strategy that may need to be considered, and so far have not been done apart from patients with athemia caused by a stromal defect.
And I guess here in that case, no, sorry, Bodo, but I'm not an expert. I wanted to know, I guess when you introduce the thymus, you need some immunosuppressant for a long time or not. When you co-transplant the bone marrow plus the thymus, would you need a different immunosuppressant procedure, or as for bone marrow transplantation, you can get rid of immunosuppressant after the first year? I'm not an expert in bone marrow transplantation and and not in thymus transplantation. So I don't know if there is a clear answer on that, but
yeah, I don't know either. So I think there are only 2 thymus transplant centers in the world. One is in London and the other one is in South Carolina, I think. Yeah.
Okay, thanks. And we have a question from Elizabeth Ang. She asked for the average rheumatologist what initial workup or investigation would help increase the suspicion of NFkB-opathy.
Well, that's a good suggestion. And I add to this, would you do anti-interferon alpha as a typical procedure to detect this sort of immunodeficiency, or have you set up in your team this screening in all your CVID patients?
So yes, and that's because we collaborate with Paris very strongly. So if we don't find a genetic cause and we think that autoantibodies may mimic the phenotype, we send serum samples to Institut Imagine, to Tom or Paul or Anne-Puel, and to ask for autoantibodies. And I think if you have patients with hypogammaglobulinemia or CVID, you always should think of an NFkB-opathy because at least in our adult cohort, as I showed you, 1/4 of the patients, this is the defective pathway. If you are a dermatologist and have a patient with rosacea, for instance, if that rosacea is familial, it immediately would trigger genetic investigations. If that patient has rosacea and something else, let's say rosacea plus antibody deficiency, rosacea plus interstitial lung disease, it would again immediately trigger the genetic investigation. And the same is true for Behçet's disease. If you have a Behçet's disease with pyoderma gangrenosum or with something else, it always would trigger for me the suspicion of an NFkB-opathy. So it's a combination. And this is why we tried not only to develop this clinical severity score, but we need also to evaluate this 21 feature score for diagnostic purposes.
I see we have another question on, and maybe the last, on NFkB2 haploinsufficiency or gain of function. So have this patient a severe lymphopenia over time? Tom, do you have an idea whether or not this patient kept the lymphopenia over time?
So usually what we observe, at least for patients with EKB delta gain of function, is that they have a reduction in the memory T cell compartment, And in the NFkB2 haploinsufficiency, it's a reduction in terms of naive T cell. It may be that some patients have reduced have a T cell lymphopenia over time, but it's not something which is consistent over the cohort. And I'm not aware also of autoantibodies against T cells, for example, in this patient, but that's something that may be worth looking.
Okay, but definitely, I think in the workup needs to be deep immunophenotyping of the B cells to learn more about the distribution of cells in addition to this autoantibody screening. I think we are done just after 1 hour of webinar. So it was really fantastic, Tom and Bodo, to have you for this excellent session, for the discussion. And so it's recorded. You can see it again if you want to learn more about these two fascinating diseases, and send your feedback about this webinar. So thank you, everyone. So the speaker first, and also the attendees. And see you next time. Thank you very much.
Thank you, Jade and Alexandra. Bye-bye.
Bye.